And also thanks so much for this opportunity to present virtually. And yeah, I just feel sorry for not being able to be there in person. So yeah, hello everyone. I'm Qianwen Wang. I'm currently an assistant professor in the University of Minnesota. And today I want to introduce you about our research where we try to use large language models to understand how people are using visualizations in the scientific analysis. And specifically, we try to understand how people are using high-dimensional visualizations when they doing analysis in single-cell transcriptomics. And this studies is conducted with Xin Liu, who is a PhD student in Northeastern universities, and Liz Glenberg, who is associate professor in Harvard Medical School. And part of the studies are done when the first two authors are associated with Harvard Medical School. Okay, so first, we all know that visualizations play a very, very important role in the current scientific data analysis. We use it to explore the data to generate insights, to share the insights with our collaborators, and also to communicate the findings in the final publications. However, visualizations, and it's good and effective visualizations, are not easy to create. Like this is not only regarding like whether we have effective tools, but also about sometimes we don't know which type of visualizations that's actually a good candidate to communicate our patterns. And at the same time, often when we open a biology paper, we would be impressed by the qualities and amount of visualizations it presents. It is common for new beginners to wonder, how can I create visualizations like this? And at the same time, for visualization researchers like me, I'm wondering like, how can we learn from these flood of figures? And can it actually guide us in better design and development of visualization tools? So ideally, with so many high quality figures available from the existing literature, this should offer a very unique opportunities to explore and answer those questions. If we're able to conduct a comprehensive analysis of those figures, we should be able to help beginning researchers to learn from the best practice and avoid pitfalls and common mistakes. We should be also able to help visualization developers to understand the real user needs and learn, and which will inform the development of more effective visualization tools. And with the with the growing capabilities of large language models, now feels like the right moment to do these types of analysis. Of course, we cannot do the analysis for all type of data. And so we choose single-cell transcriptomics as a focused test beds, both to assess whether this large language model-based approach is feasible and to explore what kinds of insights we can uncover from visualizations in this increasingly important field. Here's a little bit background about single-cell transcriptomics. Single-cell transcriptomics goes beyond bulk analysis of tissue samples by measuring the gene expression at the level of individual cells. So it offers a very high resolution insights into the cellular heterogeneity and the dynamic progress that are often hidden in traditional sequence methods. The underlying data is also very interesting because it's like by default high-dimensional data, typically represented in the form of a cell by gene expression matrix. And so high-dimensional data visualizations is actually a well-established and studied field in the visualization research community. So these combinations are important domain field with rich visualizations and a visualization that has a clear definitions of well-established research makes this domain a very ideal test bed for what we want to do. And specifically in this study, we use existing data sets about single-cell transcriptomics literature, which include more than 1,000 papers, from which we are able to extract more than 6,000 figures. And of course, like some of them are just diagrams and some of them are high-dimensional data visualizations, which we will discuss more later. And so far, looks like we have everything we want to have for the analysis. We have a rich data set of figures from single-cell transcriptomics papers. And we have the powerful large language models, which should help us to process and reason over those figures. But of course, things are not that easy and that simple. And the first challenging, which you have probably heard in many other large language model related contexts, is hallucination. A straightforward idea to do such analysis would be we're just giving the entire PDF files to the large language models and ask it to do the analysis, just to answer which type of visualizations are used and how they are used, which questions they help to answer. But in our experiments, this often doesn't work without further prompt fine-tuning. So as the examples show here, the model often produces incorrect or irrelevant interpretations. So the first insight about his map descriptions is correct, but the second one is only mentioned about like which patterns or insights we get from this cell circle trajectories, but not mention the visualization. The third one, it actually hallucinated a visualization that is not used in that paper. And we believe this is because high-dimensional data visualizations are deeply embedded in the domain-specific narratives in those papers. So visualizations is usually not the main focus. They serve to support about logical findings. And so often the time, the visual encoding themselves are not explained very explicitly. The second challenging is the input length limitations. While it is relatively easy to use large language models to analyze like one paper at a time, there is a strict limit on how much content we can provide in a single prompt. So how can we scale this analysis up to hundreds and even thousands of papers to get a holistic view of the visualization practice is also not clear. To address those two challenges, we propose a human-in-the-loop large language model workflows. And specifically, we want to analyze how people are using visualizations in the papers so that we can understand the common analytic tasks, the typical visualizations that people are used for each task, and also what are the effective strategies and also frequent misuses. And here is an overview of the LL workflow. The key idea behind this approach is to break down the overall complicated task of large amount literature analysis into small, well-structured subtasks. And each subtask will have their own clear definitions and expectations for the input and output. And we also provide a list of well-crafted examples so that the large language models can handle it more effectively and accurately. And this approach also aligns with what we learn from existing literature. The more structured and constrained the input is, the more reliable the large language model output tends to be. And at the same time, I also incorporate several important human validations during the, I'm sorry about this, about the qualities of the workflow. So it allows us to scale up the analysis at the same time, also maintain the quality control of the results. And now let's go through each of those subtasks one by one. So the first one is to translating the original text that is about a specific biological analysis or insights to a more standard and generalizable descriptions about the data formats and analysis tasks. So why we want to do something like that? That is because among the more than 1,000 literature papers, even though they're all about single cell analysis, they might use very different terms to describe different cell types, tissues. Even when they are performing very similar types of analysis, they can use the similar visualizations. So to enable large-scale, consistent analysis, we need to abstract those specifics into more generalizable terms. So for example, here we see 3 different examples that are from different papers, but indeed they are all about analysis that item clusters are differentiated for different K-dimension values for the gene expression. So secondly, we extracted those specific visualization related concepts by analyzing those general terms. And those keywords think of how high-dimensional visualizations are being used. Again, why we want to do this subtask? That's because even after translation, the texts are still rich in analysis details. And so to make the analysis more structured and searchable for the following analysis, we extract the key analysis concepts from those texts. And we do this by first automatically extract the candidate keywords from the general terms that are translated, and then group similar terms based on the semantic meanings. So like some trajectories, developmental paths, and differentiate directions are all going to group into one single term. And this step also involves a human in the loop process where we review and refine the keywords to ensure that we are capturing the core idea accurately and consistently. And after this step, we got two group of different keywords. One is about insight relevant keywords. So like what type of insights or patterns people want to extract after doing those visualizations. And second, we got a group of data related keywords. So it's describe which types of data are got involved when people are creating those different visualizations. And those keywords gives us a structured way to reason about the visualizations and the intent people use to create those visualizations and analysis people conducted upon those visualizations. And in step 3, we're going one step further by looking into the semantic context of the visual analytics. So for instance, that means we not just say that the term clusters appears, but also try to understand what visualizations is doing with the term cluster. So that the people identify a new clusters from the data set, or do people try to understand the relationships between different clusters? And to do that, we begin by identifying figures that are linked to each of the keywords we have identified before, and then interpret the surrounding context to infer what kind of visual reasoning is being applied. And to guide this process, we will provide a set list of context categories which are derived from the menu analysis of a small simple set, let's say 10 papers. And then we use large language models to match each of the examples to this list and also propose new categories if none of the existing categories apply. Okay, now let's take a look at the results, like what do we get from our analysis. So the table on the left shows the context category of each insight relevant keywords. And those context categories are related to the data related keywords. So when people say trajectories, it's related to the analysis on the item movements, on the change of dimension values, or like on the comparison of different trajectories. On the right top upside plots, we see the frequency of each visualization, reasoning types, and also say like how often they happen together with each other, which highlights the complexities of the analysis. And in the bottom bar charts, we can see the usage of different visualization types and the relationship between different insights. Due to time limits, I will just try to report a subset of our analysis, but more detail of our analysis are available in our papers, along with a web-based browser where you can explore all these papers that we have analyzed, and also now there's different texts we use to filter and analyze those papers. So, and here are a subset of the results that I want to show today. So first, we organize our results around the 4 common analysis tasks: the clustering, the trajectories, the distributions, and the correlations. And for each of them, we summarize the typical visualization strategies and the patterns of use. And again, more details are available. So due to the time limits, I will just maybe show clustering very quickly. So you can see here, so for clustering-based analysis, which is actually the most dominating analysis task in our visualization corpus. So we can see the dominating visualizations are scatter plots and a heat map, which is not surprising, but also the combinations of those two. And so some tasks that we are supporting the next clusterings of items, but also some other analysis tasks. Like sometimes people try to identify the clusters of dimension values to identify like similar genes with similar expression patterns, and not that well supported. And a similar way to do that for the trajectories, for the distributions, and for the correlations. Also, there's some common practice and that we found very interesting during our analysis. So first, people, even though all the visualizations used in our analysis are standard visualizations, line chart, scatter plot, heat map, we also see some novel looking visualizations by combining different visualizations together to create a multi-view representations of the same data so that the people can have more perspectives in one single visualization. And we also see that another interesting observation is related to the use of dimension reduction techniques, such as UMAP or t-SNE. So because those methods do not guarantee the global structures are preserved, so we see sometimes researchers using an actual visual validation step. So for example, here, they first use color to encode the pursuit time to ensure that this temporal structure is preserved in those global space after dimension reduction. And then they use this heat map to conduct analysis of the cell type changes during the pursuit time as a trajectory analysis. We also see something that might be able to get improved, and the first thing, or also most common thing, is about the use of color encoding. Color is often used to encode the different cell types in scatter plots and to such as a UMAP. However, we know that humans can only reliably distinguish about 8 to 12 papers in complex visualizations. But often we see scatter plot use can be more than 30 unique labels to indicate cell types, which this practice, even though it's very common, can be confusing. And maybe alternative encodings, instead of using color, we use contour or densities, might provide some better, better alternatives to better showing the different clusters and the overlapping. Okay, there are some evaluate. So do I need to stop here?
You have 1 minute or less now. If you'd like a question, you should wrap up.
Okay, so I will just wrap up. So of course we do quantitative and qualitative evaluations to understand how good this workflow is and there are also definitely some limitations we have in terms of the human efforts requires and reliabilities and also the domain generalizabilities. Okay, so in conclusion, we have a multi-step human in the loop LLM workflows for analyzing visualization usage and specifically high-dimensional visualization in single-cell literature. And use that we have a deep understanding of how high-dimensional visualizations are used instead of like classic analysis tasks and representative visualizations, and also how the common usage strategies and some misuse scenarios that might be improved. And with that, that's the end of my presentation.
Fantastic. So, Qianwen, we will maybe be able to take a question during the changeover. But I think for anybody who has any long or deeper questions to Qianwen, please contact her directly. Also, you can ask questions on the Juno app as well. So is there a really, really quick question right now whilst we do the changeover, or are you just happy for Thomas to take the floor next?
Yeah, maybe Thomas can go. Yeah, and you should feel free to reach out to me if you have like, any other questions via email or like, via the Juno platform.
Okay, so thank you very much, Qianwen.
Yeah, thank you.